collect. So there's a little bit of discussion here in the chat room.
Let's look at this video.
Roger. Want me to come in?
Negative. Stand by one.
Yeah.
I would like to go off stick. Can you tilt up for me, co-pilot?
Sure. Grabbing the camera. Tilting up.
And then just come in video on
Coming in the
either.
base here?
Yeah.
Okay.
I just
Tilt up a little bit.
I can't be up and out.
It looks like there was a pycnogonid on top of that.
Stand by video.
On top of that chrysogorgid in the back.
Come out a little bit, just while we get
Just
set
hold
up here.
there. Okay.
Coming in. Well,
That'll pass.
that'll be gone soon.
Tilting down.
Yeah, and this gives you a little bit of appreciation as to how unbelievable and how talented our video engineers are and our ROV pilots just getting these phenomenal images under these challenging conditions. Sometimes current is blowing, and it's just remarkable. Of course, the technology is also quite remarkable, but it still requires a lot of skill to get these images and get them in focus, and it's quite remarkable.
That's all right.
Probably
Probably rack.
Yeah.
What's in the middle there? Of the one in the background.
There?
Yeah, I just saw.
You're looking for an associate?
Hard to
No, it
say.
just looked like an enemy or something in the middle.
Yeah,
Oh,
but I
yeah,
think
I
if you
see it.
pan left, there is a pycnogonid or sea spider. It might be just slightly left and down. Not
We'll
much.
try. You can go down on the camera.
Copy. Oop, too much.
Come wide video or partial. See if we can find the sea spider. I don't see it.
Yeah, there was on that lower branch, but it might be hidden now behind the rock.
Oh, okay.
Down there.
You can get this one in the foreground if you want.
Yeah, I think those are tube worms on the rock, the Sabellida. Those would be polycute
man.
tube worms.
That's
And,
a
they're-
fried ticket ride.
yeah, he's
He'd
looking
break
like
his
one of
arm.
those windsocks. One of those things you would see at car dealerships. So it's just
For those of you who didn't have a camera on Dan, he did a perfect impression of the windsock just now.
Wish I saw that.
Alright, come
Yeah,
a little
come-
wide here. Maybe real wide.
Yeah, I
we-
think we can go full wide.
Yeah. There you go. Full wide.
Copy. Okay.
Alright, there's your rotator back power.
Thank you, co-pilot. Thanks for the help.
No problem. Looks like it's pretty tough to hold position there.
Well, I'm off stick right now.
Yeah.
It rotates with the tether. You're good.
And that's again a nice view of just how much life there is down here, and how much large life. There's
Poor rock box keeps blowing open.
there's a lot of big animals. Coming up on this massive glass sponge. It's going to be in a genus Poliopogon, part of the family Pharonematidae. characterized by these hook-shaped attachment to substrate, and I think we just saw a large anemone to our right.
Still a local high.
Do we want to try to image the enemy?
Yeah,
I
maybe
see it.
quick snap zoom, I think, maybe for
Sure.
video.
I still don't have it.
I see it in the manip cam, right there.
There
Yeah.
it is.
I mean, it's fairly common, but we haven't seen one today.
Thanks for getting lined up again
And
a
so
little
that's
better.
going to be in the genus Exosolactus.
Any chance we could take a look at the sponge that's off to the right? It's just to the right of the screen.
Yep. Well,
Okay, video.
good.
Coming in. And holding there.
And I was wrong, so this is it's
Negative.
not a Exosolactus. I would think this is a Hormathiid.
Yeah, go ahead, co-pilot.
And so yeah, the request is for a sponge next to us.
Okay. Did you not want to image this anymore? We good here?
Yep, I think that's a good view. Thank you.
Come on, video.
Wide eye.
Sponge to the right. Thanks, co-pilot.
That one up ahead, Chris?
Right to the right? Oh, there's two Chris's.
Yeah.
Yeah, that one, I think.
Yeah, perfect. Yeah, to the right and bottom of the screen, just about to leave the screen.
Yeah. The one that knows a lot about sponges.
I think. Yeah, so Andrea, I think that was a Hormathiid and the genus Phaleactus. From afar, it looked like this Exosolactus, but it definitely was not that.
Oh, we've got some time.
Ship has stopped.
Yeah. You may as well just take the rotator now, co-pilot.
Copy. Rotating down onto the sponge.
Just hold there.
Copy.
Okay, go ahead and video.
Coming in.
And come down, co-pilot.
Hey Daniel, this is Andrea. I was just talking to Chris here about how large some of the sponges are in this region. Yesterday noted quite a few large sponges today in the Polyapagon, and we haven't really seen that in the Atlantic and the Gulf of Mexico. Nothing that I'm aware of, at least. So it's just an interesting observation about these large sponges in this region.
Can go in more.
Yeah, and that goes back to what we were saying
Yeah,
earlier,
go
though. This is the first time we've been able to say
looks like
something about
maybe,
distributions of some of these major groups in different ocean basins and across major ocean basins, is that as the Okeanos Explorer is going to more places that
Tilt
are
up slightly.
further spaced
Come on.
apart, we finally start to make some sense
Right
about
there.
what a distribution patterns might be.
Yeah, I don't
I don't have
know.
any idea.
Video.
Chris, any idea here with this sponge?
Did you see the movie Alien? I'm still collating. I don't think I know the sponge.
What?
Excellent, that makes me feel better too. There's
Try
a-
it again. See if there's some forms
I
or
don't.
anything.
I don't see it in the guide.
Lost you on stereos.
Yeah, but it's
You wanna-
very
Before
interesting
you come all the
what
way out, you want to tilt up?
Andrea's
video?
mentioning
Or,
that
co-pilot,
different
and
parts.
Keep that an enemy in the background.
Alright, coming down a little bit. Oh,
And that is a Corallimorpharian. So yeah, that's the
How we
first
doing
time
with
we've
serious?
seen a Corellian Morpharian on today's dive. So they are
Nav.
not anemones, but they're
Ship's been stopped for a while.
um,
You want me to pull the ship back?
they're in a separate
No, I'm just asking you.
order
how we're
of
looking.
hexacorals.
They stopped earlier
Sister
too, and
to
it
the
blew 50
anemones
meters
and
afterwards, so.
sister
Yeah,
to
it's
zoanthids and
drifting slowly.
scleractinian corals.
Okay, go by video.
Yeah.
Thanks.
I'd say we're good to continue on the same
Mike.
heading or even a little bit
Thank
more
you, Gideon.
southeast.
I got the camera back. Thank you, Copilot.
Copy.
An urchin.
And depth is
Yeah.
1,865 meters.
You want to go ahead partial?
See if there's any interest.
Oh, and that's our first urchin.
Oh, sorry.
And I believe this is genus Sparosoma.
Want to keep coming?
Yep, and Chris agrees, and he has gone one step further and provided a species ID. That's Sparosoma obscurum.
Yeah, go
Okay.
ahead.
Video. I think that's.
So
They
D2
got a
should be
species
like
that's coming
Alright.
right.
right?
Yeah. Over here, it looks like you can see his light pool in your fisheye. But I would use your tilt cam, yeah, that guy, to go locate him.
So again, for our shore-based scientists and viewers that might have joined us later, we have about 30 or so minutes left on today's dive. So we'll try to cover a little bit of ground here for the remainder of the dive. So we landed at 2,015 meters. We're now at 1,865. So we just climbed about 150 meters of vertical relief and some very interesting communities here. Really dense aggregations of.
You notice when you get into this more cobbly type of substrate, you're losing a lot of animals, and then when it starts picking up and you're getting larger boulders and other formations, then the animals pick up. I think there's enough current here where a lot of these corals just can't stay upright on one of those small cobbles, and just like last year, they may get knocked over and die.
Yeah. It's a great observation, yeah, and something that we see that it's not just that these animals need favorable flow, they also need favorable substrate. They need a kind of stable environment that they can attach to, and
Yes, can you repeat that move, please?
Yeah, you know, Chris, I can't be absolutely sure just because we haven't really taken a look over the edge.
Good copy,
But
thank you, Bridge.
So
Brian and
Move
I
is
have
still
been
272.
discussing this this
Go get a
landslide
quick
scarp
snap
that
on
just
that
wraps
rock,
around
so
the
you'll bring
entire
that kind of rust
landslide
discoloration
deposit,
on it.
and it's
Can you
incredibly
admire-
steep.
What's that
Assuming
video?
that incredibly
These
steep
rocks
also
that have
means
this little
incredibly
discoloration on them, just center left.
sheer
Yeah,
cliffs
just like the one
with
we've seen.
very solid substrate, and also it
See
looks
a
like
bunch
the
of these.
currents are kind of going transverse to the
Grabs
ridges. There might be high density coral
I don't
and
know if that's
sponge
the
beds just on the
geo
other
or
side of the
bio on
ridge
it.
too.
Okay, go ahead. I'll try to hold it as much as I
Looks like
can.
biology.
Hold there. Yeah.
Coming out a little bit.
Yeah, definitely. We're obviously still trying to figure this all out. What really makes the best type of habitat for these guys? So the more information and more observations we get, we may eventually really nail it down.
Yeah, so the landslide scarp is not incredibly
Yeah, we're
steep
good.
where we're diving
Thank you.
deeper currently, but if you take a look at the new multi-beam that we collected last night, you can see some just sheer cliffs that extend all the way around this horseshoe-shaped valley, and marking the headwall scarp. So if this was a
30
mass
minutes
wasting
remaining.
event that
30
was
30
catastrophic
minutes.
in one big event, it was most certainly a more recent event. So the the ridges haven't had some time to reestablish their oversteepened cliffs and turn into talus and rubble slopes. So just on the other side of the ridge might be some high-density coral beds too, but like I said, we haven't gone there, and it's only speculation at this point. But just seeing how high-density populations, or seeing the high-density populations that we've seen so far in today's dive,
see what's on any of these.
I'm sure that they also exist on other aspects of this terrain as well.
Okay, go ahead, co-pilot, and go ahead on in video.
Coming in.
We got some time.
And that's a good illustration there. You see the skeleton exposed on those two branches to the left, and then to the right you have tissues on top of the skeleton. You see how different that looks. So that tissue on the bottom that is covering a skeleton, it's called the coenenchyme. And then you see the polyps protruding.
That little,
Pardon?
tilt up a little bit.
Yes.
And so just like
Sorry,
us,
I thought you were asking me to tilt up.
humans, corals have the skeletons in the inside. And yeah, you can see there on that exposed piece of skeleton, there's some hydroids, so all those kind of long, finely branching things that are coming off, so the corals, even when they die, provide habitat for things.
Transitional areas between.
And it looks like there's perhaps that white squat lobster is also perched on top of it, and there's a shrimp.
So right center frame.
Yes. Or we can just mosey around it if you want to come up some more.
Yeah, you want to tilt up? Just
And the coral to the bottom, that's a pink coral.
You can keep coming up if you want. Let me come out a bit.
Hey Daniel, Sandra, I'm just getting confused on these pink corals that are resembling Paragorgia to me.
Tilt down.
But you just said that was a pink coral as in hemicorallium?
I believe so, yeah, as we zoomed in, but yeah, it's definitely a pink coral.
Yeah.
a bunch of them.
Yeah, you see multiple.
Remember it. And one.
Go ahead.
All right. I think we got most of it.
All right, thanks.
Okay.
Yeah, I agree.
And coming out.
Copy.
But yeah, it's a good point, Andrea. Yeah, they're sometimes very difficult to tell them apart, and particularly from afar. There's slight differences in coloration.
There's
But
a Xenophore, yeah. Coming at us.
as you see more and more of them, like the boundaries between them become, especially when you see them from afar.
Rotate it back, Bob.
Okay, thank you. You didn't know you were going to be full-time cameraman, huh?
Move complete.
So it still looks to me like the local high is along our current heading. Right now I'm a little bit south of west. 254, say 270. So if we want to keep along with local high, we can just keep on this heading 270, 260.
Sounds good to me.
Bridge, ROV nav. Repeat the move, please. 30 meters, 270, 0.2 knots.
And there's a little bit of discussion in the chat room about these tall colonies and
The one with yellow.
that they're found
Yeah.
on tall boulders and
Oh, boy.
stability
The
of
one
the
we did stop
environment
and
and
look
of
at had bio on it, but I don't think we have to go back.
yeah,
and
but
look at that
absolutely,
one.
there's,
Oh, we don't
there's
have-
some absolutely
Oh, it's
massive
further
anyway, so-
colonies that
Okay.
we've imaged today. A few colonies that are over 2 meters high in height. One of the primnoids
Yeah,
that we
right
tried
there.
to sample earlier, yeah,
On the
was
left?
over 2
It's
meters
just
across,
center
so
now, on the
taller
ledge
than an
by
adult
itself.
human being. And
Yeah, you can come in on that.
Right above laser.
Yeah.
Coming in.
They just hold
It
there.
just doesn't look like the others. Maybe it's bio on it.
It's an urchin.
yeah, good
Yeah.
spot.
So we can get the urchin.
Yeah.
Yeah.
Stand by.
I believe that's going to be in the
On the wrong
genus
side,
isn't it?
Cenopadina. Take a closer look here.
I think coming out.
Yeah.
That's full line.
Be pushed up, but I don't even have any Z-by. It's 3.
And excellent. Yeah, it looks like
Let's
Chris
go back.
agrees
Try to get that.
with that ID.
We're going to do a quick shift change over here, pilot.
Copy.
And that's fine.
Can we zoom video?
Zooming.
Just trying to get the urchin a little closer. Look at the urchin.
Want me to grab it, pilot?
No.
Okay.
Following your lead.
Yeah, just hold there. Okay. Just cut my video. Thanks.
Yeah, up into port.
Yeah, it's so light. Or not light, but it's just weird. It's usually the other way around.
So yeah, very massive chrysogorgid colonies here. That's all of these enormous ones, these planar fans. And yeah, from afar they don't look like chrysogorgia, but then as soon as you
Okay.
zoom in, you see those
Snap zoom
characteristic
video?
polyps.
I'm gonna rotate around it.
And there's a good view of that.
No. Thank you.
Can we see the fuzziness of the polyps?
So yeah, we have about 20 minutes remaining on today's
Yeah, come on.
dive, and yeah, so for tomorrow and as far as planning, we are going to be diving on the seamount that is about 80 miles
Okay, you can take it now,
southwest of
co-pilot.
us, and that's really our only option because we need to get closer
And
further down south
ahead and
because
video,
then
yeah.
Friday we need to leave and transit to down to Kwajalein, and so yeah, we are going to be mapping. We're going to make a straight beeline after the ROV comes up on deck, transiting down to the seamount, and then we'll have to map it. So there are no multi-beam
Sorry,
maps
video.
for
Trying.
that seamount, and so we'll map that and
See, it's not easy.
then pick our dive site accordingly.
Dan, while we're looking at this, can I make a comment?
Yes, please.
I just find it interesting, given all of the space that is available here on the substrate, that these two fans are pressed against one another, because you would think that one would be interfering with the other in terms of obtaining food out of the water that's flowing by. It's just an interesting observation that these two are both fairly large, so they've presumably been there for a while, but both are surviving just fine.
Copy.
Yeah, that's a very interesting observation. Yeah, there's a lot of
Yeah, I can tilt down a little bit, please.
available substrate around it. So yeah, it's where these colonies start and where the larvae settle. And it might be something unique
So
about.
I see from this angle that the bases of the colonies are not right next to one another. So it may have been that one is growing faster than the other. So, for example, that Chrysogorgia perhaps is putting out that's the white one that's on the left. Maybe is putting out a branch that is now gone behind the Paragorgia, or the Paragorgia grew up after the Chrysogorgia. I'm not sure. I don't know the relative growth rates of those two. It's just kind of interesting to observe.
Yeah, no,
Come back
I
to your
appreciate
center
for
screen, and
sharing that thought, and yeah, that is
figure out what the sonar targets are.
again, unfortunately for a
Just
lot.
adds to what we've been saying quite frequently, that a lot of these observations that we make on these Okeanos Explorer dives just lead us to ask more questions and kind of get all the scientists excited about coming back and thinking of ways that we can do experiments or set up other observations that we can test some of these ideas.
Yeah, that's a great point, and that's exactly what this program is about, is trying to do exploration and get some preliminary data that can be used to develop some hypotheses. But yeah, we go to all these places for the very first time, and most times we don't know exactly what we're going to be finding. And so you can't really develop hypotheses before you go.
Back to 270.
Oh, and there's our first rodent Iridogorgias. And that's a great spot.
Looks like we are moving a little bit closer.
And then hopefully,
tight spiral. The last sweep, it was at 25 meters.
yeah, a lot of these expeditions hopefully has of course, the hopes that it leads to some follow-up work in the future.
Did you say
and,
something, video?
I said that's a tight.
yeah, as you mentioned, there's a lot of stuff to follow up on now that we have some preliminary data and observations. Again,
Maybe a snap zoom on this in the background. It's like
A
half
coral.
Yeah.
and yeah, Scott is again putting your comment in the chat room, observing that these are 3 colonies clustered together, and why is that? That some when they are the same species, then that could lead you to believe that something about how these things disperse, that the larvae don't disperse far enough, that they settle close to the adult. But when it's the 2, like an image before where we had 2 of different species clustered up next to each other, then it might be something that particular rock or that little microenvironment was particularly suitable for the larvae to settle. So yeah, it's all interesting questions
One of
to
the
explore.
reasons I wrote that, Daniel, saying that there were 3, and I was a little bit not necessarily surprised, but I put an exclamation point there, was because I don't think we've seen any other Rodan or Iridogorgia on the dive, and then suddenly there's 3 together.
Yeah, and then this has been brought up before, is that, and in some cases we see a lot of what appears to be a perfectly suitable habitat, perfectly suitable conditions, and there are no animals there, and it might be just because they haven't a chance to get there, and that's just a matter of time, and then once they get there, there's a source for new recruits in the future. So yeah, great point, and yeah, thanks for sharing that, Scott.
It's a nice perspective on Sirius. All the corals down the edge.
And yeah, that's Jonathan just left the room, but I agree that this looks like some pillow lavas. If we're looking at some pillows here,
It's steep too.
our expert geologist has just returned and has some.
It kind of looks like we might be looking finally at some more intact lava flows and pillow lavas with these more rounded shapes and kind of some nice radial fractured patterns. So left of center of the screen kind of looks like there might be a toe of a lava, pillow lava with these subtle yet almost clear fracture lines that seem to be going towards the center, what the old lava toe would have been.
I'll keep coming in. On these walls, it's a little more important to get it pretty much dead on to our
as we started going up this ridge a little bit more, the slope is steadily increasing, so giving us a little bit more clear look into the actual rock outcrops themselves that haven't been buried by talus debris or any kind of rocky rubble.
Or can we rotate the upper swing arms more forward? Twist them?
Right now, they're as far forward as I can go. We tried to adjust that
Out
earlier.
that way?
Yeah.
Copy.
Is that a fish down there? Or is that another coral?
We're not
That's a
seeing
coral,
the
yeah. Sorry.
Should I pull the lower swing arms in? Since we're going up a
I think they're okay.
copy.
Hey, Daniel and Jonathan, so the video's quite a bit better right now, and I was just curious if something out there has changed besides the some fade finishing. Is the sea conditions dropped a little bit, or because it's noticeably improved?
Yeah, I can't answer that. We haven't looked outside all day.
From video standpoint, the ship is still rolling and rocking about the same. Satellite is tracking as accurately as ever, which is very superb. This entire expedition.
Very far. If I can put it in.
Thanks, Roland.
Say again now?
Sure.
Final ship move.
Sounds good.
Bridge, ROV nav. Yes, one final move, please. Same as the last. 30 meters, 270, 0.2.
There's a bright white dot to that coral that's leftmost on your screen.
Roland, I'm gonna try to work my way starboard here. I need to get back to center. I'm just fighting it, and that's because my lateral is faulted. Okay.
Yeah, and again, one of these, you can
Let me
see
know
where
if
the
you see
current
another
is
one of
coming
those
from,
there,
and then you can see
Copy
all the
that.
fans are all oriented all in the same direction and all kind of perfectly oriented to intercept the current flow and the particles that might be coming in the current flow.
It's actually any of those super white
Dead
dots.
center, yeah. You want to do a snap from here?
It may just be old
Thank you,
fence or
Bridge.
a
Good call.
sponge.
Yeah, that's what it looks like.
Sponge.
You know, for as old as this seamount is, supposedly,
Standby.
it just doesn't really look like there's much heavy manganese crust on these, so this could
Let's just
potentially
let those
be
go,
a
Roland.
little bit more evidence for the recent
Down to the
event that I was describing
Yeah.
earlier that Brian and I have been talking about, where the this landslide that has been causing a little bit of headwall erosion to occur along these ridge lines is more or less a recent event and exposing the surface of these rocks in a more modern type of in geological terms period. So manganese crust does not have as long to
Copy that.
begin precipitation on the surface of the rocks.
That's off bottom time. You guys only need to bring Sirius about half a turn. So got a pretty easy setup aside from swing arms.
Copy that.
So we got about 10 minutes remaining on our dive, and we're at a depth of 1,835 meters.
Yeah, and it's really been a spectacular dive. I've really enjoyed it, and really we're harvesting the fruits of the hard labor that our mapping team did overnight. They stayed out all night, and yeah, this was definitely a great selection for a dive site. And hopefully we can repeat the same thing tomorrow. So tomorrow we'll be diving also on a feature that has not been mapped yet. So we'll head down there and then map it, and then hopefully be able to see some interesting topographical features that we can dive on.
Those whip things sort of look like cattails on the left.
But it's very exciting. That's something that we've done throughout this expedition, as we've gone on a few of these seamounts that hadn't been mapped at all, or just partially mapped, and you go first, you put the sonars over it, get an idea of what the bottom looks like,
Can't tell if it's a coral
and
or a
then
sponge.
you really get an idea of what the bottom looks like with the
You
ROV.
can snap if you want. It's like a coral.
Yeah, and then
Science, we've seen these before today, right? Well, actually, we've seen these before today, right?
yeah, not up close, but yeah,
Do you want to get up close for one?
we can.
Yeah, if we've seen them
come on.
throughout, but we haven't
Can you come on?
imaged it, we should get at least one good copy.
Yeah, they're tough to ID.
This will be our last shot then. Co-pilot, you want to start to think about which way I'm going to go to get set up to recover?
Copy. So, Nav, seems like I'm going to rotate a half turn.
See if we can get it up in the light.
To port.
So I don't know if I'm completely right in what I'm looking at,
And then
so I've been trying to convince
pilot
myself over the
will
past couple minutes, but it looks like there's
rotate
this arching
half
type
turn
of shape
starboard.
to those rocks that are just now out of the top of the
If you look at it,
screen.
he's just about lined up with the aft of the ship.
Yeah.
So you're the only one that should need to make a turn.
Started
So
Hey,
now
there's
that
one plus
we're
Okay, go
looking
ahead,
at
Roland.
it dead center, it
Okay, got
kind of looks like those rocks have like an
Okay, so
arching
I'll just
fracture
swing around
texture
to-
to them, which might suggest that this was the
would
cooling
suggest
shell
swing
of
it now,
a
just
pillow
little
lava
bit,
at
not
one
the full
point, but
turn, but just swing a little bit now.
it's not
to see-
completely
till you see that
obvious,
turn
and I may just
option,
be tricking
Okay.
myself
Confirm.
into seeing what I would like to see.
which direction you're going to go,
Yeah, and
and that it should remove,
yeah,
should see it decrease both
this
turns.
is a primnoid octocoral, so not a bamboo. And Chris is providing Candidella gigantea as a possible ID.
So I see it.
It's definitely not an arella.
I don't know, Dan, because it looks like you got 5 polyps coming out of each point, and I think
So you get around your tether, you're probably going to want pilot to back up towards you before you.
sorry.
maybe it's 4 coming out of each point on a lot of them, but I think it's too many for Candidella gigantea.
Yeah.
Your tether.
Full screen, if you can stay
Yeah, you're right.
can't hear
Just a
video.
fine, delicate.
Let's see if we can get full
polyps.
screen if you can restabilize.
Yeah.
Move is complete.
But yeah, those have 3.
Can you tilt up for me, co-pilot?
Copy.
Just get the full specimen. Yeah, there we go. Maybe up just another hair. Alright, here we go. There we go.
Co-pilot, it's time for you to start bringing in the swing arms.
Copy.
He is on the wall, so make sure you clear your uppers before
Yeah,
you.
whenever
bring
you're
them in.
clear video, just let me know.
Copy that. You can pull off as I pull out.
So yeah, we're unfortunately
That'd be very graceful.
pretty
Try.
close to out of bottom
Okay,
time
maybe
here.
look
It
at-
has been a
Full line.
Copy.
Okay,
Uh,
I'm giving you
wonderful
rotator
dive.
pilot.
Copy that. Thank you.
very first dive,
So make
very
sure
first
you
surveys of this
you know
seamount.
the motion on the lower swing arms, right?
So,
Final
Take-
depth here is
take the full right or full
1,838
left,
meters.
yep,
Before you bring them to the stow position.
okay.
So use that camera on your left hand to move
I
around
wanna,
and get visual. Either that
thank
camera
all
or
of
the one
our
you use.
viewers for joining us. We will have another dive for you tomorrow.
Yeah, I'll probably give you a little bit better view.
So thank you so much for joining us. For our shore-based scientists, I don't know if there isn't really much we can discuss for the pre-dive planning, because we're going to be diving on a completely unmapped feature. So we'll have to map that overnight and then come up with a plan in the morning.
Just for lowers.
Okay.
Lowers can hurt things if you don't do it in the right order.
Copy.
All right, Dan, John, you guys did a great job picking today's dive site, so I fully trust you to pick us something equally impressive for tomorrow.
All right.
Well, we will relay that to our mapping team and put the pressure on them, so they better find us something to pick that looks just as impressive as today, huh?
Yep.
Yeah, we'll see. We'll try our very best. Of course, the features have to be there. But yeah, thanks so much to all of our shore-based scientists for joining in and providing absolutely valuable feedback and comments and IDs. Big mahalo to everyone, and then big thanks to all of our viewers, and of course to our pilots, videographers, and the rest of the crew.
So now, Stowe?
Yep, thank you guys, and a special thank you to you, Brian Boston, and teaming up with me to discuss that awkward geology feature that we saw, those fractures along the entire day. It was nice to discuss some of those thoughts with you, so you take care, buddy.
Okay, that's full Stowe.
My pleasure, Jonathan.
Point. Thank you. Okay, so now the starboard lower, full to the right. Still trying to get some movement.
If you want, you can go left just a hair, and then push it full right to see movement.
Okay.
That'll make you feel better. Pretty sure it's all right though, but you're fully right.
All
So
right.
there you go. There's the left. Now just push it hard to the right. Yep.
Okay, so now Stowe. Oh, there we go.
And then your uppers are fine. You can do those in either order.
Okay, let me get a little bit better view.
You're gonna have to use mini Zeus to do that, or ask your pilot to.
I was going to get a look with Sirius's HD too.
Like a tighter shot from Sirius.
Sure. It's going to be a little bit hard to keep it. Okay, so then I think that's good video. Thank you. So those, they don't matter. Just right and left.
Oh, it can tilt,
Okay.
so we'll go all the way in and then all the way up.
Okay. All right. So starboard upper stowing.
The HD2 gives you the best view of that.
Okay.
I just don't know which joystick it is.
That should be this one. Which monitor do I have it on video? Oh, I see. Starboard upper. Yeah, starboard upper smear.
That's not the starboard upper. You need to point up, forward, and straight up.
So,
Come left. Yep. Pan left and pull the stick towards you.
thank you.
Keep going left. You're both of those. So they'll be. Those are your mains. You need to keep coming.
Is that good?
Yep. Now if you stow them, you should see them just.
Yeah, let's
one pair there.
let's stow them, and then we'll worry about the orientation on the way up.
Okay.
Does that work? So just stow both.
So the starboard is already stowed.
Yep.
So we'll go to stow to port. Okay, stowing upper. Port, swim up. Okay.
Okay. And what did you guys figure out? You need to come to the.
I need to rotate, and that should
Yeah.
be it. Right?
Okay.
So should I come about?
You're gonna turn right into your tether if you do that.
Yeah. So let me back up,
Okay.
and you come up.
All right. I'm hauling in.
You can haul in a little quicker. There you go. Not that fast. Let's make our delta like let's make our delta like 15 or 20.
Copy.
Something in there.
Okay. I'm marking you guys as ascent.
Copy that.
And dropping target.
Copy.
So pilot, that's
Okay.
20.
So you should be clear now
ROV.
Yeah, I see the tether underneath.
to turn.
Okay. So turning to port.
Copy that.
Yeah, it's underneath me.
Whoa. That's okay. At least you have eyes on it.
Yeah.
Polykeet thing? What was that?
Okay, pilot.
Okay, hold there.
Zeroed out.
Yeah. I'm way down.
Do you want me to pay out?
No.